Imagine your body is hosting the most exclusive party ever. Now, every drug is trying to get in. Some walk in confidently, and some get pushed through the side gate, and some just stand outside, confused. Today, we're inside the party entrance, the biological membrane, and how the drugs sneak through it. Let's look at the gate itself. This is not a simple wall. It's a double-layered gate, about 100 angstrom thick, made of phospholipids and cholesterol. Now picture this. The outer surface are friendly. They are hydrophilic. And the inside is fatty, a lipid coat which is hydrophobic. Basically, outside is welcoming, but inside, picky. This is why only certain drugs can pass easily. Now, this gate is not empty. It's full of moving parts. And there are proteins floating around freely, like staff members inside the party. Some are extrinsic proteins hanging around the surface. Some are intrinsic proteins going all the way through the gate. And these intrinsic ones, they create tiny channels like secret tunnels. On the surface, you also have glycoproteins and glycolipids, basically sugar decorations on the gate. They help in recognition, like checking who belongs and who doesn't. Now, apart from the main gate, there are also tiny gaps between the cells called paracellular space. Think of them like small gaps between two doors. Not everyone can fit though, but some can sneak in. Now remember, passive diffusion is like walking straight into the party. If you're lipid friendly and in the right form, you just enter. No question asked. But filtration, it is completely different. How? Filtration is like being pushed through a side gate with holes. No one is checking your personality, your vibe, your chemistry. But there is only one question. Are you small enough? Now visualize this. There is a fence with holes, and suddenly a crowd pushed from behind. Now, if you are small, you will slip through. But if you're big, you'd get stuck. Yeah? Now, that's exactly what happens in the body. Drugs pass through the aqueous pores or paracellular space, driven by pressure, like a push. It could be hydrostatic pressure or osmotic pressure. No role of lipid solubility, no role of pH, only size and pressure. Now, here is an interesting part. Most cells, like intestinal cells and RBCs, have very tiny pores, so large drugs cannot pass. But capillaries, they have large pores, except in the brain, the BBB, the blood-brain barrier. That's high security, people. So many drugs, even large ones, can pass through easily. So in capillaries, drug movement depends on blood flow, not on lipid solubility, not on pH, just how fast the blood is moving. So now you know passive diffusion is like walking into the party. If you have invitation, you get in. Filtration is like getting pushed through a side gate. And the membrane, it's not just a wall. It's a smart, dynamic, and slightly dramatic gatekeeper. That's all for the day, people. Thank you for listening.